My mission is to understand the relationship between genes, health, and the environment, and human wellness, what we call today precision medicine.
Cancer precision simulation is about making sense of what happened for the individual. It's about two things, really. It's about the individual, but it's about precision.
We are very grateful for the support of a multi-year investment from the Advancing Healthier Wisconsin Endowment, which are allowing us to push the frontiers of genomic beyond where they were ever before. The current standard for genomic diagnosis is primarily based on the information obtained from the DNA sequence. We call this method of diagnostic 2D genomics because it is based on an interpretation that is linear on the sequence of the DNA and your genes. This linear coding in the genes make molecules in 3D that works like nanomachines.
Clinical genetics has in a way been in flatland. We have on the bottom of the screen is a representation of a gene, and it's a linear sequence. And at the bottom you can see these colored ticks. These are sites where there's been a difference in different people, a genetic variation. And if we see the same variation in another patient, well, that's maybe what happened, but we really don't have a good sense of what the gene is. So I want to take the patient, a representation of the patient that has a variation of this gene, and put it not in a land of 2D, but in a land of 3D, where we can better understand that it's not this sequence of ATCG that causes the disease, it's dysfunction of that protein complex that's in this case an ion transporter. And if we can calculate what happened to that protein in 3D, for a reference, and then what happened in the patient, now we can get a sense of what's different and how it's altered and what it means for the patient.
Our own methods now represent the gene product in 3D and assigned to this gene not only the structure in 3D, but also the motions that it has to do in order to give rise to a particular function.
Each patient may have an alteration in different parts of this little nanomachine, and what is altered and how really could have ramifications for how we try to help them later.
And furthering the impact of the Mellowes Center is working with the Milwaukee Regional Medical Campus and beyond, including hospital partners, the Clinical Translational Science Institute of Wisconsin, and the Office of Clinical Trials to make new genomic-based medicine available to the population of Milwaukee.
The HW endowment grant has helped us to build a platform. So what we've been able to do takes our experience in precision oncology, takes our experience in the undiagnosed diseases, and we've been able to build a process that is supporting both of them. And that's something that's difficult to fund through some of the traditional venues that are very disease-focused.
It is allowing us to develop, optimize, and implement new methods for genomic diagnostic. Furthermore, to create new tools and methodologies in system biology that are now available to all the investigators that seek to improve the health of individuals in Wisconsin. Thus, the impact of this work goes from gene to molecules, to cell to humans, to the community. 3D genomics is a transformational approach that is calling attention worldwide.